Well, good afternoon. I'm George Daly. I'm the Dean of Harvard Medical School, and I'm delighted to welcome all of you to this special symposium to mark the launch of the Harvard Medical School Translator and Ideation Hub. Thank you very much for joining us today. I, of course, wish we could be celebrating together in person rather than virtually, but rest assured that day will come. Today is indeed a celebration. But more importantly, it's also an invitation. One of the traditional strengths of Harvard Medical School, and part of what has made me so proud to be a member of this community now for almost 4 decades, is our profound commitment to curiosity-driven fundamental science. Our dedication to illuminating the mechanisms that underlie human health and disease has generated myriad life-changing medicines. And it's allowed us to train leaders and innovators who've shaped the landscape of biomedicine in Boston and beyond. As Dean of HMS, one of my foremost priorities is to cultivate and enhance the impact of our community's research, and to do so by facilitating the advancement of transformative science from our labs to the clinic in service to patients. Now, this was not a decision made on my own whim. When I was first appointed dean, my leadership team and I conducted a survey of hundreds of investigators across Harvard and at our affiliated hospitals, and we documented an astonishing level of interest throughout our community. Now, in response, we launched the Therapeutics Initiative. Now, we wanted to remove as many obstacles as possible and provide our community with the resources needed to propel promising ideas into new medicines and therapies. Our priority is to pursue therapies based on their potential impact on patients and society as a whole. And this includes prioritizing the medical needs of people of color, other traditionally underserved populations, which the recent COVID crisis has clearly taught us bear a disproportionate burden. We wanted to also craft new educational paradigms that are focused on experiential learning to help train the future inventors of the next life-altering therapies. And importantly, to instill a passion for therapeutics in those who will build the future of biomedicine. And we wanted to create a community without boundary, one driven by creative, rigorous, collaborative science and a genuine desire to solve the most abstruse and recalcitrant challenges to human health. And these goals with the establishment of the Therapeutics Initiative are well within our grasp. In recent months, I point out our school's response to the COVID-19 pandemic, which is highlighted by our anchoring of the Massachusetts Consortium on Pathogen Readiness, MassCPR. And this revealed just how formidable we can be when traditional barriers between researchers and institutions are knocked down, and we unite to confront a daunting foe. Now, we have no intention of returning to business as usual after the pandemic is controlled. Most importantly, this initiative has been fueled by the historic gift from the Blavatnik Family Foundation. Consequently, we have invested deeply in research infrastructure, in graduate therapeutics education, in systems pharmacology and regulatory science, in computational biomedicine, and in the ongoing construction of the Blavatnik Harvard Life Lab here in Longwood. Now, we've provided and will continue to provide crucial funding for promising research projects across Harvard and our hospital affiliates through programs like QFaster, the Dean's Innovation Grants, and a program that serves as an exemplar for what we are working to achieve, the Blavatnik Therapeutics Challenge Awards. Now, I am thrilled that Len Blavatnik has joined us today for our symposium, because it is my privilege to announce today the inaugural recipients of the Blavatnik Therapeutics Challenge Awards, and we will be hearing from Len in the closing comments. These awards provide timely and critical support for 5 projects that are on the cusp of clinical translation, each one having the potential to transform the lives of patients through powerful, creative approaches based on rigorous fundamental science. If I can have the slide that shows the award winners. The teams are led by, number 1, Dan Bauer, one of my colleagues, an associate professor of pediatrics at Boston Children's, is working on a gene editing approach to treat severe congenital neutropenia. This is a rare genetic disease that renders patients vulnerable to life-threatening infections beginning in the first few months of life. And this treatment has the potential to be curative. Elliot Chaikoff, the Johnson & Johnson Professor of Surgery at the Beth Israel Deaconess, who is developing new therapeutic approaches to prevent cancer-associated venous thromboembolism, the second leading cause of death in cancer patients. Christian Ferran, the Lewis Thomas Professor of Surgery at the Beth Israel Deaconess, who is working to provide type 1 diabetes patients with long-lasting glycemic control through gene therapy, an approach that may serve as one of the first alternatives to insulin therapy. Stephen Greenberg, Professor of Neurology at the Brigham and Women's Hospital, who is developing novel monoclonal antibodies to target the cellular mechanisms that drive severe asthma and other inflammatory diseases. And finally, Peter Park, Professor of Biomedical Informatics in the Blavatnik Institute at Harvard Medical School, who aims to restore the function of a gene implicated in the devastating neurodegenerative disease called frontotemporal dementia. And this will be using antisense oligonucleotides. To all these awardees, congratulations. The Blavatnik Therapeutic Challenge Awards represent one of several key components of the Therapeutics Initiative that are now in place to support our community. This substantive foundation has been established thanks to the leadership of Tim Mitchison, Carolyn Shamu, Peter Sorger, David Golan, David Huang, Ifat Ruben Bejarano, and their teams. And this spring, a key cornerstone was put in place when Mark Namchuk joined us as Executive Director of Therapeutics Translation. Mark brings with him over 2 decades of drug discovery and development experience at the highest levels of industry. And I am truly excited for the future of our therapeutics community under his leadership. Today, we're going to learn more about the vision of these leaders for the initiative, particularly with the launch of the Ideation Hub and the Translator. These 2 components span the breadth of translational science, and together will help researchers formulate actionable ideas from basic science, and then transform those ideas into something that can make a true impact in the clinic. We will learn about their efforts to build upon our strengths in scientific education and fundamental discovery, and to build a culture of collaboration and community that will advance Harvard Medical School's mission of alleviating suffering and improving health and well-being for all. Today is truly your invitation to join this community. Now, I'd like to introduce Tim Mitchison, the director of the iHub. And Tim, the virtual podium is yours.
Thank you very much, George. And I really want to echo George's word of welcome. I'm thrilled that so many people have tuned into this virtual symposium. I'm also sad we aren't doing it in person. But to really be specific about the welcome and the invitation, really to reiterate George's words, the Ideation Hub is very much about building community and helping everyone, people, scientists at all stages of the career pipeline develop their ideas. And so if you have any ideas that you would like help with, or anything is triggered by any of the work you're going to hear today, please drop me a line or contact anyone else on the initiative, and it'll get to us. So what is the Ideation Hub? Can I have the next slide, please? So this is an outline of the whole therapeutics initiative in kind of shorthand form, and it's got a number of components. You just heard about the Therapeutic Challenge Awards and the partnerships with local institutions, and I am so thrilled that we're doing this across the Boston-Cambridge ecosystem. That's going to hugely empower everything. I'm using it to sort of situate the iHub within this larger initiative. And I stands for ideation, but if you wanted to stand for innovation, that's just fine also. Can I have the next slide, please? So the iHub activities are really broadly in two areas. Building community with the purpose of ideation and project creation. And we seek to build a diverse and inclusive community encompassing scientists and doctors at all stages of the training pipeline interested in therapeutics research, and also to tweak HMS culture a little in the direction of team-based research. And I'll talk more about that in a second. And then the iHub is also the center for the educational initiatives in therapeutics, where, as George stated, and I really want to double down, our goal is to train the next generation of leaders in therapeutics research in industry and academia, with a focus on experiential learning, and also very much a focus on the science of how medicines are made and the science of taking fundamental discovery that George alluded to and turning that into new medicines. We have some partnerships with the Harvard Business School and other parts of Harvard that are interested in the kind of business side of things, but iHub is really going to focus on the science side. And if I can have the next slide. Next slide, please. I've sort of broken down those into activities directed more at PhD students or MDs, at fellows and postdocs, and at faculty, and we'll remark on one activity for each stage of the pipeline. And although, you know, community building and ideation have been separated here from education, in everybody's mind in the Therapeutics Initiative, these are really two sides of the same coin. You know, we're going to build community through education and vice versa. And I'll touch on one initiative in each of these areas. So if I can have the next slide, please. Starting with perhaps more directed at faculty to begin with, but can certainly involve others, is the disease and technology group. And this is a broad concept. It's not a very specific template. But I want to first enunciate the hypothesis, and that's that translational research thrives when teams have diverse and complementary skills. So, you know, I think there's a sort of spoken or unspoken tradition at Harvard of one professor slightly in their own castle building a beautiful intellectual edifice, but we aren't convinced that that's the best way to really impact ultimately on the clinic. And if I can have the next slide, please. So the idea of disease and technology groups are faculty-led groups to tackle a specific disease or a new technology or diagnostic. I think we're very broad on that. Emphasizing a team-based approach, we very much hope to combine quad faculty with hospital faculty, clinicians, but also perhaps some less traditional participants, industry experts, disease advocates. Whatever team really makes sense to tackle a challenging problem collectively. We imagine these teams, and I'll show you our first two examples in the next slide, but we imagine funding them through philanthropy, through grants, and very much through industry collaborations as well. And we want to support them with TIFs, which is a new kind of postdoc. And the next slide, please. So the first two, we're incredibly excited about the first two groups that have launched in Lyme's disease, led by Mark Namchuk, Isaac Chu, and Jun Hu, who are two young professors in immunology, to tackle this notorious medical problem in the Northeast of Lyme disease. And then the second one is a larger project to tackle COVID-19, which is a multi-pronged collaboration with AbbVie, the big pharma company, and has multi-PI teams in 4 distinct areas. And this one I have a close-up and personal experience because I'm part of the host targets team led by Pam Silver. And I just, as a personal one, I can't tell you how much fun. There's 4 professors on this team. We meet regularly on Zoom, and we're hashing out ideas for our collaboration with AbbVie. It's just so exciting to be combined in new ways with professors from across the whole school. And for both the Lyme's disease and COVID, we're in the middle of TIF recruitment. If any of the students or postdocs on the call are interested in positions, please contact me by any means. And the last is your idea here. I mean, iHub is committed to fostering ideation and team building. So for people who have ideas and either know how to pursue them and want help, or don't precisely know how to pursue them, we would love to hear from you. The next slide. Moving a little early in the training pipeline, I want to talk about therapeutic innovation fellows. Here, the hypothesis is a new kind of postdoctoral position will drive therapeutics projects forward and support faculty. They'll also provide a career path sought by many PhD students where it's difficult for people to find training right now. And the graphic just symbolizes that we know that our PhD students, but also across the country, are now seeking many different kind of careers, not just professor. And the next slide. So this is a postdoctoral training program, a 2-year program leading to careers in industry and also management tracks in academia. It's not primarily for people who want to be a professor, although that's certainly not ruled out. The trainees will be mentored by faculty leading disease groups, but will find co-mentors matched to their career goals, many of whom won't be professors. It's going to be 80%-ish experiential learning supporting one of these disease and technology groups. We sort of imagine 2 tracks, a kind of ideation, management, fundraising track, depending on the need of the groups, and also potentially an experimental science track. And then there's also going to be a skills and networking curriculum. And the first 2 TIFs we're actually recruiting now will support the Lyme and COVID. But if people are interested either in those disease areas or this concept in general, by all means reach out to us. We hope to build a small community of these fellows in the next couple of years. Next slide, please. And I'm coming towards the end of my time. Let me briefly mention the Therapeutics Graduate Program, which is the faculty directors, David Galan, who's the PI of the T32, and myself, and we're ably, Catherine Debray plays a key role. And this is it's a certificate program open to all Hills and HMS PhD students. We particularly want to thank Fujifilm for a gift that helped support this program. But it's a dedicated curriculum. It's a way to find community outside your parent program if you're particularly interested in the science of drug discovery. But it's for students who are interested in professorships as much as industry positions. So it's certainly not industry specific. And really the most innovative portion of this, and the thing that's probably most significant for the students, is the required internship. And this is a national model in experiential learning. This is not the only PhD program that you can do an internship. Actually, most of the programs allowed that. It's the only program that really requires one, which we find helps students negotiate with their faculty advisors. And this has not added time to degree. It's been a huge growth opportunity for students. I can't tell you how great it's been, as well as an important networking opportunity. The next slide is just a statement of mission. This is really here to just show a picture of people, because the TGP and really all the iHub activities are about bringing people together. And this is a wonderful community of PhD students and mentors to really create a diverse and inclusive community interested in therapeutics research. And my last slide, please. Oh, sorry, second last slide. This is just where the interns have gone. So a little more than half of them have gone to research and industry, but the others are spread out in a whole bunch of other interesting internships experiences, typically in the middle of their PhDs. And the last slide, I won't try and read this out, I'm sorry, but a recording of this whole talk is available if people are interested. These are some quotes from students we asked about their internship experience. For example, let me I can read the middle one. The TG plea has played an impactful role in my PhD experience and given me the skills and opportunities to be a strong applicant for jobs straight out of my PhD. So there's a student who presumably isn't looking for a professorship, but about half our students do. And the last student actually contributed an article, Jessica Savers contributed an article to a Nature Family journal, 5 reasons to do an internship during your PhD. So we think this is really great. And if any early stage students are on the call and want more information, please drop me or Catherine DeBray a line. So thank you.
Thank you, Tim. So my name is Ifat Rubin Bejarano. I'm the Director of Translational Research Programs here at HMS. And I would like to tell you a little bit about QFaster. I actually, before coming to HMS, I co-founded a company, a biotech company based on my postdoctoral research at the Whitehead Institute at MIT. And so I'm trying to use that experience to help professors also take their basic research and translate it towards commercialization. So QFaster stands for Quadrangle Fund for Advancing and Seeding Translational Research. And what we really do, what the fund does, is to identify, support, and expedite translational research. Next slide, please. So the fund has been in existence for 5 years now, and we like to think, I hope by the end of this presentation to convince you that it has been quite successful. I have some outcomes at the end. But what we really try to do is to fund what we believe is exciting, leading edge, early research on therapeutics and diagnostics. The people who are eligible to apply are quad professors, but what we found is that really many of them tend to collaborate with affiliates, with professors at affiliated hospitals and some other institutions. And some of our best projects are actually collaborative. We focus on the impact on human health and of course also on commercial potential. Next slide, please. So a little bit about the approach. You know, we realized that we have an amazing set of professors here at HMS. They are the best in their fields. They are mostly doing basic research, and they are expert grant writers. So they really don't need help writing grants. But what we realized that is really different is with translational grants, that the way you write them is different. So if you write an NIH grant and you can propose to do one thing and end up following, as Dean Daly mentioned earlier, follow your curiosity, and that led to many very exciting discoveries. In translational grants, you really try to form a plan that would provide a proof of certain concepts, certain therapeutic, to show that it's doing something. So what we end up really doing is working very closely with quad faculty to crystallize their ideas, to create a milestone-based plan, and whenever needed, to help them craft pre-proposals and full proposals. And then once proposals are selected and in the execution stage, we really are there to meet with them and provide scientific advice and troubleshoot, because always, you know, it's good to have a plan, but plans change and you always have to pivot and make changes and amend as things progress. We're also there to provide help with contracting if it makes sense to work with a contract research organization, and to provide advice on when to file intellectual property and on early interactions with investors. And especially the last part, we do in close contact with the Office of Technology Development at Harvard. Next slide, please. So we have 2 types of awards, pilot awards and development awards. The pilots are $50,000 a year, and the development 250, typically over 2 years. The pilot awards are really to support a critical experiment, maybe an essay development, something that needs to be done so you could provide, maybe be more attractive for later stage funding. And the development awards are a bit more advanced, although everything is really early. and that if successful would be attractive to a larger funding resource. Next slide, please. We try to be really open to any modalities, any therapeutic areas. You can see on the right, in terms of therapeutic areas, in the last 5 years, we've funded many projects in neurobiology, neurodegenerative diseases, infectious diseases, cancer, but many other areas. And on the left side, you can see the modalities, a lot of small molecules, but also substantial portion of biologic drugs, gene therapy, oligonucleotide, and so on and so forth. And we're really open to all of them. Next slide, please. So to give you some example of what projects we support, I'm going to actually announce the financial year 21 development awards. Next slide, please. So the first one is Karen Adelman, Splicing Modulators for Therapeutic Impact in Cohesin Mutant Cancers. So this is a project that actually started as a pilot and now is getting the development grant. Karen and her team is collaborating with Zuzana Todhova from Dana-Farber, and they discovered that cohesin mutations are common in some cancers. The QFaster funding will enable them to develop, together with Peter Park, also from HMS Therapeutics, to target those targets that she identified. And if this all goes well, this will be really new therapeutics targeting novel targets in cancer. Next slide, please. The second project is James Chu, development of an HIV-1 vaccine. James also is collaborating with Bing Chen from Children's Hospital, and they use a new technology to present the HIV envelope glycoprotein in a membrane environment. The QFaster funding will enable the team to test this formulation in animal models. And maybe I'll just add that, the idea here is that because they have a new technology, maybe the presentation will be more efficient, and that will yield an HIV vaccine, which we all know is needed. Next slide, please. The third one is Doug Melton, a novel biologic for the treatment of diabetes. Doug and his team discovered the novel peptide that can reduce blood glucose levels without causing hypoglycemia, which we know is a big problem. And the QFaster funds will be used to test the activity of a recombinant peptide in mice. And this project is actually co-funded with the Blavatnik Biomedical Accelerator. Next slide, please. Mike Springer, development of a diagnostic for broad respiratory panel. This project also was a pilot that is now receiving a development grant. Over the pilot, Mike and his team developed a quantitative, cheap, rapid, sensitive, and selective method for detecting influenza virus. And now he's actually broadening this up and adapting this to a broad respiratory diagnostic panel of viruses and bacteria. The idea is that many times they all lead to very similar symptoms. So having a diagnostic that would differentiate and characterize exactly what people have could be very useful. I would also mention that when COVID hit, actually, Mike used the work that he did with flu and very quickly adapted to detecting SARS-CoV-2, and that resulted in spinning out a diagnostic company. Next slide, please. Last but not least, from the development grants is from Peter Sorger and Jeff Way, Biologics for the treatment of pain. Peter, Jeff, and their team are developing protein therapeutics for pain with a novel mechanism based on snake venoms. And the QFaster support will allow the development of molecules with tunable selectivity and potency, which would be great. Next slide, please. I'll just highlight the 3 pilot awards that we're giving this year. Mark Albers, target identification of neuroprotective kinase inhibitors for Alzheimer's and related dementias. David Ginty, identifying novel therapeutic targets for treating pain and chronic itch. And this one also is in a collaboration with the Blavatnik Biomedical Accelerator. And Mo Fuliao, structure-guided discovery of novel antibiotics inhibiting bacterial cell wall formation. And so you can see from those awards that they tend to be a bit earlier, but if successful, they can apply for the development grant. Next slide, please. So just to summarize, this is the impact we've had so far and some outcomes. In 5 years, we funded 38 projects, 14 of them were pilots and 24 development grants. We've had 43% of quad faculty apply, and we would love to get this number even higher. So if any of you have ideas, I would love to hear from you. In terms of outcomes, 2 companies started based on QFASR projects. Another project moved into Lab 1636, which is a collaboration with Deerfield. 1 project, the last one that Dean Daly mentioned earlier, that received a BTC award, was a QFASR project, so we're very excited. There are also 29 patents and 8 papers that are associated with QFASR projects, and over 62 million in follow-on funding, which corresponds to 9.2x on the amount of the QFASR awards. Last slide. So the new RFP for the new round is actually going to be released next month. So please contact me. My email is here. I'm sure you can find it. I'll be happy to talk to you about your science. Next to speak is Mark Numchak. He's Executive Director of Therapeutics Translation at Harvard Medical School. Mark.
Thank you, Ifat. First, thank you to everyone who's joining us today. Welcome. I'd like to add my welcome. I'm going to spend some time introducing you to the HMS Translator and then wrap together some of the things that you've heard from George Daly, from Tim Mitchison, and from Ifat to give you a sense of how these things fit together within the broader Therapeutics Initiative. Next slide, please. So one of the things that was highlighted in the Dean's initial comments, and I think is really a worthwhile place to start the conversation, is to remember that the impact of truly important medicines can be extraordinary on human health and well-being. Here are just 2 examples on the left, one that gets quoted often, but potentially not often enough, which was the miraculous outcome of the introduction of antibiotics as a treatment. Shown in the left-hand panel, you can see in the lower part of the panel in red, the decrease in mortality as a consequence of infection. And I think notably, you can see the spike at the left-hand part of the curve coinciding with World War II, where the case was that battlefield fatalities were more often the consequence of infection after a wound, more so than the initial trauma itself. And the impact that antibiotics have had on our world going forward has truly been extraordinary. On the right, a more contemporary example that overlaps a bit with my time at Vertex, and really, I think, brings home this idea of how an initial discovery can have an extraordinary impact when translated into a medicine on human health. And this is a stack of different innovations that came along over a period of time, moving forward the treatment for patients with cystic fibrosis. And I think it's noteworthy to remember that the initial gene for cystic fibrosis was found in a collaboration between the University of Toronto and the University of Michigan in 1989. And that initial discovery within an academic institution fueled decades of research, which are now leading to medications which have the promise to give patients with cystic fibrosis a long and very healthy life. Those sorts of important medications are the goals of the Therapeutics Initiative based on the extraordinary science that's going on at HMS and within the broader Harvard community. Next slide, please. So in trying to put together the therapeutics program and fitting it into HMS, these were the 3 guideposts that we used. First and foremost, that anything that we do from the point of view of the science of therapeutics would be conducted at the highest level of scientific rigor. This is a community that collectively, I believe, has 46 Nobel Prizes. We have to make sure that what we do scientifically in the pursuit of a medicine is of the scientific caliber required by our community. Secondly, and George Daly touched on this in his opening remarks, HMS has this extraordinary ability to be in the center of the community, not just academically, but the biotech community. And that allows us to think about this initiative as being borderless science, interacting with different institutions, different disciplines, with industry to surround an initial insight with the capability to transit it towards a medicine of tomorrow. And then finally, touching on comments that George and Tim had made, I think that we're trying to create a new conduit within our community where our fundamental science can have a direct impact on human health. First, through the education of potential future leaders in the community, both academically and in industry, whose mission will be to be the inventors of the medicines of tomorrow. And I think secondly, equally importantly, to surround the ideas in our community, our faculty, our students, our postdocs, with the means to take that initial idea and move it forward. Next slide, please. A cornerstone and home for the initiative will be the Blavatnik Harvard Life Lab at Longwood, which will hopefully start construction in the next year. For those of you that are familiar with the geography of the Longwood area, it will be on Building C on the first floor, which is on the Harvard Quad or the HMS Quad. The intention by both the design of the building, how it's integrated into the community, will be that this is a gathering point for the entire community around Harvard that's interested in therapeutics research, welcoming projects not just from within HMS, but collaborations from across the communities. Equally, we hope it will be the gathering point for numerous different fellows programs that are more oriented toward therapeutics, one of which, the Therapeutics Innovation Fellows Program that Tim introduced you to, but creating this community where like-minded people interested in creating the medicines of tomorrow can meet, exchange ideas, and learn from each other. Helping with that learning environment will be this collision of academic science and a mixing pot with people that we are having join us from industry with extraordinary experience in drug discovery. Again, looking at this through the lens of the science of how a drug is discovered. Next slide, please. The pieces that in any initiative, the first thing one needs to look to are the people that you're going to bring on board, critical and a cornerstone to any successful initiative. And we wanted to surround ideas that arise from our community with support, both from a material standpoint, but also with advice and a chance to learn how drug discovery is done, and to pass that on to our community. 3 components within the Therapeutics Initiative are listed here. The first are the senior therapeutic scientists. These are industry scientists with 20, 30 years of experience in the art of making a drug or a therapeutic, who have a real interest in being involved in starting projects with us here in the community, and equally an interest in passing on what they've learned to the people that would be involved in the project. Part of this experiential learning factor that Tim had brought in, taking people from industry and giving the opportunity for students, postdocs, faculty to learn from what they've seen and how they've been able to move drugs forward in their past lives. Secondly, scientific project managers. You met one a moment ago, Ifat. These are our eyes and ears in the community, having a chance to go out into the broader Longwood area, hear from different faculty about what their ideas might be, and do anything from help write that initial translational grant all the way through to helping to execute on some of these larger and more complex plans, like we see with the Blavatnik Therapeutic Challenge Awards. And then finally, to highlight the Therapeutic Innovation Fellows, the glue that will go out into the community and really help build groups of scientists across disciplines, across institutions, that are willing to collaborate to tackle really difficult translational problems. And that multidisciplinary research is the middle of everything that we're trying to accomplish. Next slide, please. The translator will be set up to support a spectrum of different projects based on their stage. Certainly, when the Life Lab is built, we'll be able to house early startup companies that are getting the pieces together that will allow them to eventually exit as an independent company or to transact in another way. Somewhat different than a traditional incubator, we plan to have earlier projects pre-company creation also exist within the Life Lab and be supported by the translator. This is a case where, for example, if you use an example from the QFaster work that Ifad introduced you to, where we can be involved in helping to design the scientific architecture of a project, moving it from that initial idea through the experiments, which will really allow it to grow towards being a medicine of tomorrow, and start that in very early in the process, all the way through to when that process might lead to an exit. Finally, and a part that I'm really excited about is, as I've spent more time in the HMS community, a lot of the folks that are here are so busy with all of the things that brought them into academic science, it's difficult to make a full commitment to a translational idea, even when it might be absolutely superb. And here, given that we'll be able to have industry leaders join us with vast amounts of experience leading therapeutics projects, as well as the capabilities we built within project management, we'll be able to support a project in a much fuller way to be able to take that idea and move it forward, even when the investigator may not have the time to take a leading role. To be clear, this project still belongs to the investigator. Our role here is to find ideas in the community, surround them with capabilities, and move them in a robust fashion towards the point where they can begin to move to external organizations to grow on a path towards becoming a medicine. Next slide, please. Tim Mitchison introduced this slide a little earlier on. It's a busy slide, but the cornerstone in the center of everything we're going to do are these collaborative projects with therapeutic or translational potential, shown in the line in blue. In the upper left-hand corner, you can see a variety of different institutions. This is not a comprehensive list, but these are the institutions that we've been collaborating with thus far, either on projects or in different initiatives. And my ambition would be that if we do a talk like this next year, there'll be so many symbols in that corner that you won't be able to recognize the actual individual institutions. Supporting the initiative on the bottom of the slide are different components that you've been introduced to. A, the HMS Therapeutics Translator, which will be able to provide expertise in therapeutics research and development, outsourcing support, project management support, and also the Blavatnik Harvard Life Lab at Longwood, physical space where these teams can aggregate and work together. Tim Mitchison has introduced you to the iHub, which it plays a cornerstone role in the educational portions of the Therapeutics Initiative, both through the Therapeutic Graduate Program, but through the disease and technology groups and the TIFs, the ability to generate new projects that could potentially enter the translator, but also to create these communities of collaborative scientific endeavor between different institutions and disciplines. Shown on the right-hand side of the slide are other folks that would be very valuable contributors and collaborators within the Therapeutics Initiative. For example, the Laboratory of Systems Pharmacology is an interdisciplinary institute here at HMS that's already doing extraordinary work, both in generating new targets and new ideas for therapeutic projects, but I think also importantly in experimenting in new ways that therapeutics could be discovered, potentially accelerating the pace and, importantly, the fidelity of early research that takes an idea and moves it towards medicine. For some of our projects, that innovation might need to come from a different view of how we approach the regulatory science. And the Center for Regulatory Science, the MIT Harvard Center for Regulatory Science, is a great collaborator in that regard. And finally, another initiative being set up by my colleague, Robert Gentleman, the Center for Computational Biomedicine, which has also been generally supported by Blavatnik Gift, is trying to harness the power of big data across our community and could be applied in many ways, whether that be AI approaches in the initial optimization of a molecule, or potentially figuring out where you might want to go therapeutically with it. This virtuous cycle of being able to harvest ideas from many places and being able to take those different disciplines, combine them, I think will be a powerful way to move therapeutics forward, not just for the entity itself, but to continually improve the means by which therapeutics are discovered. Next slide, please. The support structure for this work has also evolved, and parts of this have already been introduced in previous talks. The translator And on the top, we'll be able to provide support at all phases of a project, from inception all the way through to a potential exit. Early funding for these projects can come forward in QFaster grants, where EFOD introduced you to the idea of really an initial grant to take that first step to see if the idea has translational potential. Our colleagues over at OTD run the Blavatnik Biomedical Accelerator, which funds projects at about the same stage, all the way through to ones that are more mature. And it was announced by the dean earlier on, we have the Blavatnik Therapeutic Challenge Awards, which are $1 million awards, allowing projects to move really closely towards that initial point of entering the clinic. And that kind of support is really critical to getting the true translational potential out of these sorts of programs. Corporate collaborations can certainly play a huge role in what we're doing. And finally, once we've got the Life Lab at Longwood, I think it allows us to think about how long we incubate these projects internally to bring into play a whole series of potential external partners. Exiting a little earlier to potentially create a company with our partners in venture, and in some cases, potentially keeping that project a little longer to allow it to mature to the point that it might transact and go directly into pharma or biotech. Again, another conduit by which that medicine can come to life. Next slide, please. Here's an example from the MassCPR. It's a program I was involved in setting up the screening infrastructure to support therapeutics development for the MassCPR at the height of the COVID outbreak, which we started working on in March and April. I add the example really to take home the point that George Daly touched on, which COVID created an opportunity to collaborate together in ways that we really had not experimented with before, between academic institutions and between academia and industry. Here, it was a joining of multiple institutions to take advantage of unique skill sets and abilities across that allowed us to knit together the support structure. In this particular case, we were trying to be able to support therapeutic development from a broad set of different constituencies, both academic and industrial. That we partnered with the Broad Institute to do compound handling, to be able to take the compounds in, plate them, and distribute them, which they had a terrific infrastructure for. Biochemical screening of targets for SARS-CoV-2 was conducted at the ICCBL facility at HMS, whereas screening against live virus was conducted in the lab of Rob Davey at the National Emerging Infectious Disease Laboratory at BU. And that effort allowed us to support initial interrogation of a number of molecules from small companies to decide whether they would be willing to start clinical investigation for those molecules against SARS-CoV-2, and also to support a number of optimization efforts across the community, not just at HMS, but at numerous affiliated hospitals. And certainly one highlight was the data that was generated by the initiative, in part supported PIK5 inhibitors from Verge Genomics entering clinical development for treatment of COVID-19. Next slide, please. A next extrapolation of putting this theory into practice was a recently signed agreement between HMS and AbbVie, a 3-year, $30 million collaboration aimed at emerging pathogens. And it hits on these themes of collaborative research and the ability to work across boundaries. HMS is the lead organization on the academic side for the Alliance, but thus far in the 4 research areas that we've put together teams, there are 16 investigators from a number of the different affiliated hospitals, as well as HMS and BU on the academic side. But importantly, AbbVie scientists are integrated into each of the research projects, which really allows us to integrate the benefits of excellent academic fundamental science and integrate that with first-class drug discovery and virology experience from a company like AbbVie. And I'm very excited to see what we can accomplish. Lastly, the small molecule programs that have been started within that initiative that I pointed to on the previous slide have become part of the ongoing initiative with AbbVie, taking those initial discoveries and being able to push them towards potential therapeutics for SARS-CoV-2 and other coronaviruses in the future. Next slide, please. We've already introduced you to the Blavatnik Therapeutic Challenge Awards. It's an example of the same principle. While we will be helping with the project management and some of the therapeutics-oriented guidance for the projects, these are awards open to the entire Longwood community. And in fact, 4 of the 5 awards went to the affiliated hospitals and 1 to HMS. And as Tim had highlighted, we're hoping in the near future to start our Lyme program, again, taking advantage of this idea of building community using a TIF to integrate different labs from across the community. The RFP will also be a RFP that will go out to HMS and the affiliates. And for the first time, a collaboration will integrate the external education capabilities at Harvard to really help a broader audience understand progress in research in the field of Lyme. Next slide, please. So our 3 goals are simple. 1, to be able to provide experience and resources at the highest level that the extraordinary fundamental science that we're doing here at HMS and other institutions around Harvard can be coupled with extraordinary translational science. Secondly, to do this in as multidisciplinary and multi-institute a means possible. They surround a great idea with all possible means of being successful. And finally, as Tim highlighted, and something I'm very passionate about, train and mentor the inventors of tomorrow's medicine, preparing them for a career in academic drug discovery or in industry. Potentially the most valuable thing this initiative could produce are scientists who tomorrow will go out and invent something extraordinarily important to human health. Next slide, please. So I'm going to close by thanking you again for joining us, thanking the Blavatnik family for the extraordinary generosity of this, the gift that has set in motion this initiative, and to tell you we're just at the beginning. We're interested in working with the broader community. We're really excited about where this will go in the future, and we'll hope to be working with you on that future in a very collaborative and open way. If you've got an idea of where you could go with the medicine, we'd love to hear from you. And with that, I'm going to call a close to the first session. We're going to have a break until 2:10. And also that we will be reconvening with what I think is going to be a really exciting part of the program, that we will be hearing lightning talks from some of our therapeutics graduate students. And we will see you at 2:10 to start that off, and that will be led by my colleague Catherine DeBril. See you in a few minutes. Thank you very much. Okay, why don't we get started? It's my pleasure to introduce our panel, and we are hopefully going to have one last panelist joining us in a moment. He has been frantically emailing us back and forth. Okay. First of all, thank you again for those of you who continue to be on the line. Our panel is tackling the heady issue of the future of where medicines are going to go, both in terms of what we should be paying attention to in terms of the future of medicine, but equally importantly, how do we begin to think or reimagine the role of academia in the creation of those future medicines. And so what I'm going to do to start off is go around the circle and have the panelists introduce themselves. And maybe I'll start out with Kevin, if I could.
Hi, everyone. Thrilled to be here. Thanks so much for having me. Kevin Bitterman. I'm a partner at Atlas Venture, based here in the greater Boston area. Been at Atlas for about 3.5 years, spent about a dozen years at another local venture firm before, or after I should say, finishing graduate work at Harvard Medical School. Great to be here.
Great. And Linda, it looks like we can see you now. Maybe Linda, if you could introduce yourself next.
Hi, thanks, Mark. And it's really great to be here. Thanks so much for inviting me. I'm Linda Avey, and I am currently working on a startup, excuse me, where we are automating healthcare and virtualizing healthcare to try to take as much of the load off as possible off busy doctors, particularly in developing markets where there's a shortage, an extreme shortage of trained physicians. And prior to that, I was a co-founder of 23andMe here in the Bay Area.
And JC.
Hi, it's a pleasure to be here. JC Gutierrez. I'm the head of drug discovery at a pharmaceutical company called AbbVie. We have responsibility for developing drugs in neuroscience, oncology, immunology, virology. Is a broad area from idea to proof of concept in humans. Over the last 25 years, I've been sometimes trying to go deep into a given mechanism and leading a company, a small company. Sometimes I've been in a broader positions in other companies like Pfizer or others, just leading drug discovery always. And I had the pleasure to start my career at Harvard Medical School as junior faculty. So it's great to be here. Thank you.
And our fourth panelist is Tim Springer, who I saw momentarily was trying to log in. I don't know if Tim's been able to join us, but I hopefully will momentarily. Tim, are you there? Okay, I don't think he's joined the Hollywood Squares. So what we'll do is introduce Tim in a moment, but maybe in the interest of time, kick off the panel. You know, the first question I wanted to take to the panel is really to give us your sense from your point of view about where the future of therapeutics and medicines is going. Part of the Therapeutics Initiative and part of the blessing of being at a academic institution is we can take a very long view about where we want to go. So maybe to frame the question, as you look out from where you're at and you think about where therapeutics and medicines are going over the next decade, what would you advocate? What would you pay attention to? And maybe if I could start out, Linda, with you to give us some thoughts on that topic.
Yeah, and it's really exciting that you guys are taking this long-term of a focus because I think it does enable the kinds of projects and studies that maybe wouldn't be possible in a company setting. And one of the things that I've always been interested in is how long that we've been using these disease labels. Say, for instance, Parkinson's disease, we call it that. And yet, I think most people have this feeling that there are subtypes, and we're starting to see those subtypes emerging. And some of the subtypes may have more of a genetic component, and others might have more an environmental component. And I think we really need to continue to spend time on this recategorization of disease and get it more fine-tuned, which then I think will match the individuals much more as we try to head and continue to head to a precision medicine way of delivering healthcare. And the other thing that I believe is really important is that we take a systems approach at the same time. When we look at a patient, we can't just hone in on one organ or just a handful of symptoms. We really need to look holistically at what is going on overall in their health. And of course, we're seeing this in cancer. We now have types of tumors, and we can type them because we actually have material that we can interrogate, but it's not so easy in other conditions where maybe what we should do is start monitoring patients more frequently, getting more data around what's going on a daily basis with them, and use that information then to really guide in the treatment and the assignment of treatments to patients. And then the thing that I do tend to harp on, if other people have heard me at other presentations, is that I think the elephant in the room is our food and what we eat. And it is something that continues to be ignored by the greater society, really. I think we're starting to see more focus on it in the healthcare system. But food is medicine. It is something that can greatly impact our health. And I think if there would be a way for your program to start to monitor that and take that into account, especially as people are being treated, then I think it's going to add a lot more knowledge around how people are responding to drugs.
Yeah, and certainly something that if you read any of the nutrition literature is becoming more prominent, and even thinking about that in the interaction of the microbiome and other growing fields. I think these things don't seem so independent anymore.
Exactly.
Kevin, maybe switching over to you, you get to see all the things in the candy store in your view, and being in venture and being in the greater Boston area. What are you seeing that excites you and that we need to pay attention to, particularly through this lens of future therapeutics?
Yeah, so Mark, I think we're at a really fascinating juncture when it comes broadly to the area of alternative modalities, what we still call alternative modalities. So not a traditional small molecule or an antibody or biologic. If you look over the last 10 years, we're now in a position where we have multiple FDA approvals of cell therapies, gene therapies, ASOs, RNAi. It's really incredible. But I think we'll look back on this time and realize that we're still very much at the dawn of this era, and we still have a lot to learn. So I think, you know, we're going to spend much of the next 10 years really consumed kind of polishing many of the rough edges that still remain in many of these modalities. And I put the work in broadly into two categories. I think, you know, some of this is work that's well underway and addressing problems and questions that are well understood. So, you know, we all see, you know, new forms of AAVs to allow repeat dosing, increased loading capacity, tissue specificity from both novel capsids and cell-specific promoters. The $4 billion acquisition of AskBio by Roche this week is a nice illustration of the interest and a lot of the work going on in this area. And, you know, a similar set of complexities limiting cell therapy still. Some of these more mundane, like CMC, scale-up manufacturability, and anyone that's worked anywhere near this field knows the criticality of those components. And then, you know, more technically challenging areas like increasing durability, moving to allogeneic sources. But I think there's a whole nother category that we're actually still learning about as an industry as we actually put more of these therapies into patients. The promise of cures of very grievous diseases, but, you know, we're also learning what really is the failure modes, broadly the therapeutic indices of these technologies. And, you know, on the more unfortunate side, we've seen recently, you know, deaths at Audentes and their high dose AAV gene therapy. Just yesterday, Ultragenyx announcing that, you know, 5 patients at their highest dose of their ASO therapy had significant and very, I think, unexpected neurological SAEs, and cell therapy as well is still, you know, quite limited in this capacity. So, you know, while we see plenty of toxicities with traditional small molecules and biologics, I think we have a lot of work to do as an industry to truly understand, you know, these therapies, particularly as it relates to the immune response to these different modalities before they can be adopted more broadly. And I think what we all ultimately hope be applied to more common diseases as well.
Yeah, and I think that larger point of we're seeing such progress in genetic and cell-based interventions at the small scale, but the difficulties in translating that can be a reproducible, exportable, usable therapeutic in the broader community. Maybe it doesn't get the same limelight, but I think we're watching this play out in real time as we think about new vaccine types and how you're going to scale that on a worldwide basis. And it is something that when you think about a medicine as opposed to a discovery, that it's definitely going to be something the community is going to have to pay attention to. JC, maybe if I could switch over to you. Give me a sense from your personal perspective. You're working at a very large pharmaceutical company. So if Kevin gets to visit candy stores, you have a large candy store. Tell me a little bit about where you think the future of therapeutics is going and places that you're paying particular attention.
Well, both Linda, with pleasure. Both Linda and Kevin highlighted important aspects of it. But I'm going to step back and say, well, if in 10 years from now, as society, we need to double remission rates in some of these diseases, because there's the only way in which we will be able to afford them, right? I mean, we're not going to be able to pay for 5% better remission rate in rheumatoid arthritis or in Crohn's or in certain cancers. So if we have to double remission rates, just as in a simplistic way, then we have in front of us a number of things have been mentioned. One is, of course, we have great new therapeutic platforms and new biology, and hopefully that plays along. But I want to focus on 2 other things. If we are not clever or lucky with biology and platforms, then what do we have? I think we have 2 things. One is combination therapies, meaning not combination therapies of one approved therapy and then a non-approved therapy like we have been doing, but rather from the beginning, combining mechanisms, dealing with an anti-inflammatory and anti-vascular drug that are both novel, or 2 antibodies, oligoclonal mixtures or several antibodies, as we know. Of course, to do that in development at the moment is financially challenging because you have implications, as you were saying, in CMC. You have to make drugs, have implications how you develop the clinical trials, how you design them. We tend to do 1 single ascending dose and 1 multiple ascending dose at a time instead of 2 at a time, have implications from the regulatory perspective. So I guess enabling combination therapies of mechanisms that rationally either because of genomics approaches or that combine well, and also that we can develop two antibodies against two different areas easily. The question is, how do we scale them up fast and affordably? That is not the disincentive. So how the implication for combo therapies is one. Second, Linda talked about it. I think if I look at my group at the moment, I would say that we take 1/3 of the resources that we have, 3,000 people, 1/3 are focused on patient segmentation. Because we know for a fact, that in any trial we have you have 30% of the patients that respond, so we cannot identify them. And of course, we have been talking about precision medicine and personalized medicine for, I don't know, since I was 20 years ago. And it's not easy because biology is complicated. But I think that to the point that has been made, single cell technologies, computational science, genomics, we have tools that, also the ability to convert some of those in assays quickly because, places like IPI are already making reagents. I think the translation of that is fundamental. So patient segmentation and combination therapies in addition to the things that my colleagues have mentioned.
Thank you, JC. And Tim, glad that you were able to join us. Sorry for all the technical difficulties if that was on our end. Maybe if you could briefly introduce yourself first, and then the question we're discussing is personal perspective on what we should be paying attention to in the therapeutic future. Say 10 years out, what are the big ideas that we need to pay attention to?
Yeah, thanks, Mark. My pleasure to be here. So I've been a professor at Harvard Medical School since 1977. And I started out in the Central Quadrangle in the Department of Pathology. I'm now at Boston Children's Hospital in the Department of Biological Chemistry and Molecular Pharmacology in the Department of Pediatrics. And I trained as a biochemist. I found the medical school a very rich environment in which to find biological questions to work on. And I found if you're a researcher, there's no reason that you can't do your research on something that's medically important, as well as, a really fundamentally interesting biological question. And so, with that principle, I've discovered the first integrin family, many of the first integrins, particularly on leukocytes. I discovered many molecules that have become drugs. And I discovered, the pathway by which leukocytes migrate out of the bloodstream into tissues in a highly selective manner. And I founded a company based on that called Leukocyte in 1993, and that was very successful. And one of its 3 drugs treats ulcerative colitis and Crohn's disease right now, and it's an antibody to an integrin that blocks immigration into mucosal sites. I sort of, retired and just focused on academics. Then I went back into drug discovery. Actually, JC here made me a resident professor at Pfizer. So I went over there once a month to review, all of the drugs in a very large portfolio, which really got my drug developer juices flowing. And then I wound up investing in more companies. And my model now is I invest and found at the same time. So, Kevin and I founded Morphic Therapeutic together. He was, we both put in an equal amount of money, and they've just announced an IND for a small molecule version of the antibody to alpha-4 beta-7, which is very exciting. And another company I founded with Phalaris, Scholar Rock, is going after antibodies to the prodomains of the TGF-beta family. And yesterday, they just announced the interim results of their phase 2 trial in spinal muscular atrophy. And they very successfully enhanced the mobility and, the ability to get around of children, under the age of 5. with that disease. So they do for the muscles what Spinraza does for the muscle. That's the nervous system side of that disease. So anyhow, yeah, so that's about me, and I'm mainly interested. Actually, a lot of my research now is really sort of more specifically directed towards interesting projects that drugs can come out of. But my perspective, and I've also founded an institute for protein innovation, which is focused on very large scale discovery of the kind of antibodies that you heard of, synthetic antibodies presented by one of the students for nanobodies earlier, but we're working on FAB fragments, and our goal is to make antibodies to lots of surface molecules that are not currently accessible to species-specific monoclonals, and really make them available to basic researchers and also, for therapeutic discovery. So when I speak to people in the pharma industry, the biotech industry, they tell me, we need more protein chemists, more protein scientists. We just can't find them, to work. I mean, we need them. And it's very interesting because there's been a dynamic whereby the strength of the genomics revolution was so strong. It's really a tsunami wave that washed over all of biomedical research. And beginning in the 1980s, everybody became molecular biologists. And it's sort of an economic thing that first of all, the learning curve in protein sciences is much steeper than it is for working with DNA or RNA. Secondly, the tools are so great. You can get your research done more quickly. You can publish your papers, more readily. So you get faster academic promotion. You get more grants from the NIH. And there's just sort of a vicious cycle where, that technology is taken over, and we have no national program, which they do have in Germany, to really fund, protein research. So, that field gets developed. So anyhow, that's one of my goals at IPI, is to strengthen protein research. And, I think that actually genomics has been pretty disappointing from my point of view. I mean, people have placed a lot of emphasis on, genes with disease associations, but what is big right now? Antibodies to PD-1, or breakthroughs. If they weren't identified by disease associations, there's almost no association. There's almost no disease association for TGF-beta, which has an incredibly important role in immuno-oncology. So, there's a lot of other ways that drugs come out, and I just think that nowadays protein scientists are very important, and I see. Not only because many of the therapeutics nowadays are proteins themselves, like antibodies or receptor decoys, et cetera, but, also because small molecules are targeted against proteins. I do see today, an incredible leap in our ability to develop small molecules from computational chemistry like Schrödinger makes available. And it's not just that if you have a structure and then you have these free energy perturbation methods, that they make available that allow you to enhance the affinity of your drug, but they also really predict what you need to do to change your absorption properties, your metabolism properties, all that. It's really amazing. And so anyhow, that's what I think is going to be important over the next 10 years.
As JC knows firsthand, in fact, our ability to be able to predict improvements in PK is now so worked into the drug discovery process that an improvement in PK may not even be a patentable invention, that we've just gotten really good at. I'm not, and by the way, Tim, you do my heart good, because I'm a biochemist, so it's good to know there are a few of us hanging together out there to try and to keep the field going. I think maybe if I could link two of the conversations together and then move to the next topic, but I think that Kevin, you and Linda started this idea around a more system thinking, and I was thinking more around cell therapy, and then linking it back to something that JC said. there's going to be this interesting collision because cell therapy is meant to be multimodal in a lot of cases. And in fact, is going to go after these different bits of the disease in different ways. So I guess a two-part question, and maybe I'll start with JC and maybe go to you, Linda. Where do we see for a complex modality like cells, where that's going to intersect with personalized medicine, which has been traditionally a little bit more reductionist? And then secondly, how do you see things like cell therapies moving into this view of system-based thinking to be able to affect the system in a more holistic way? So maybe JC, how do you see trying to implement personalized medicine in a field like cell therapy?
Yeah, I mean, it's a huge topic for the field, for medicine, right? I mean, clearly we see the potential, and the question is, how do we enable it? I think there are two components. One, I think synthetic biology and synthetic control, or whatever. I mean, control of the functions that the cell has, I think are going to be fundamental. We have seen, like with virus, like with gene therapy, right? We have seen very basic viral control, just infection and then expression, gain of function of cDNAs. Same thing with CAR T. So I think that what we are good at as a community, I think, is to arm cells, precisely as Tim was saying, because we are used to build and understand proteins that either are soluble or we put on a surface of a cell, and then there it goes, no, as CARs or as TCRTs or whatever. I think that the next thing, and we have the initial component of kill switches, that is some initial component of control. I think for cell therapy, from my perspective, to reach a broader spectrum, first of all, it's going to have to be allogeneic. Of course, autologous is great, but it's always limited. And in the past, I've been very skeptical about, as an immunologist, about our ability to cloak cells. But now I think that, again, I see, companies and efforts deleting all MHC loci and making cells and then putting specific ones, and they are stable chromosomically, even if you take large chunks. So one is allogeneic. Second, I think synthetic control of the cell, control as much as possible, not just of the warhead, but actually of the behavior of the cell. These are the two key components. I don't know how to tackle precision medicine with cell therapy, other than the obvious. You're missing something. I don't know, but it will come.
So Linda, maybe to transition to you, do we take a broader view of precision when you're thinking about an intervention like that, to your point about a more holistic view of therapy, or how do we go at it?
Well, and I'm not a cell biologist, and I'm not a developer of drugs. So from my perspective, but it seems that if we get better at finding these subtypes of disease, maybe that's the way to go about this. And I totally agree with doing combinational therapy. I think that's really going to be important, especially with novel compounds. But I think if we get better at defining disease even at a cellular level, then we're going to be in much better shape. So it's taking that holistic view of patients, being able to break it down into these subtypes, and then now let's get started on what's actually going on with the biochemistry and at the cellular level. So it's all about definition. But the thing that jumps out at me also is what's happening with the FDA. They have to be prepared for these types of drug combinations. And how can we work with the FDA sooner rather than later to get them ready for this type of approval? Because that's something that also could delay it significantly if the FDA isn't prepared for these types of submissions. So I think that's something also that I think this program really could be very good at educating and working with the FDA. My experience with them has always been very positive. So I think that's if you get started early and you start the communication, it's a long slog, but it's well worth it in the long run.
Yeah, and there is precedent for novel work in the virology field. That was a big, when I was in my Vertex days and we were doing the initial HCV comparisons, you had to do it to standard of care and through the full piece. But as more combinations became available and the biology was better established, there was much more flexibility to think aggressively about a development path. because the basic biology, the basic human biology had been established, and some of the surrogate markers made sense. So it's doable. I think the challenge of being able to do it with 2 novel therapies out of the gate is something we're definitely going to wrestle with as a community. And I do think that's going to be, if we ever got over that bump, you'd see more progress in some of these more complex diseases.
Mark, if I could add into the
Please.
cell therapy discussion, thank you. You know, I think that it's going to be huge. Its future is huge. It needs to become off the shelf, that's for sure. And you know, there's a lot of work going on to make that happen. But the limitation, I think, of the CAR cells currently is that they're all directed towards the same things that antibodies, you know, were already directed to. And, you know, we want to cure cancer. And so, you know, where's the cancer specific antigen? And I think it actually could be coming. I think that the receptors on certain tumor cells are altered in the conformational states that they inhabit. So I studied thermodynamics, and believe it or not, they had an idea for starting a new type of therapy out of thermodynamics. Who would have ever thought of that? Anyhow, you know, and when I really looked into it carefully, actually, there's already one exemplar, but I don't think they really understand how it works. It's an antibody to an epitope on EGFR that can react with cells. You know, they're just transformed cells. It's not specific for EGFR per se. And actually, Catherine Jabrell, you know, who's doing a lot of the moderating today, introduced me to a student, and he's already working on that project, you know, in my lab. So I think that there's still lots of new things, you know, to discover. So if I could and new avenues to explore in cell therapy.
Right. So if I could pivot to the second topic, which is to get your advice on how we can fashion academic industry, what should we be thinking about as a therapeutics initiative, and how do we rethink how do we build a better version of our ability to work with the outside community if in the end what we should be doing is taking the best of all worlds scientifically, combining them, and using that in the service of propagating the medicine to the people that need it as quickly as possible. So maybe if I could start with JC on that question, and maybe 1 or 2 minutes of comments from each of the panelists on this one. How do you see academic therapeutics initiatives like we're setting up fitting into the broader community, and what would your recommendations be? What would you like to see it evolve towards?
Well, I think that, again, Tim said it very well. There is great science to be done, and there is great science to be done in therapeutics. So, you know, and it's not mutually exclusive. And sometimes in the interactions between pharma and academia, or biotech and academia, it seemed it was like mutually exclusive, and there's not the case. Obviously, Tim has shown it once and again and again. And so I think that I will go back to this example that we just recently announced. Mark has been a great partner there. Harvard Medical School, AbbVie framework partnership for emergent infections. And of course, it's obvious, right? If you don't do something on emergent infections today, it's just that you don't get what is happening. So it's an obvious topic. But I think I think what is excited about it is that it's a program project type. So nothing new in terms of the model, because this has been used by our colleagues at the medical school and hospitals for many years. But in this case, it's not just money coming from, in this case, from AbbVie but it's actually capabilities. And I think that complementarity of capabilities, there are certain things that companies, either biotech companies or pharma companies, do well, and other things that they don't. And the things that do well can enable science on the academic side as well. And I think that there are some components, so this complementarity, I think this framework interactions that are not just one program, but actually one area that enables fundamental science, but also medicine, and the complementary capabilities as we were talking about, I mean, medicinal chemistry is a core competency in these companies, right? You have hundreds of talented medicinal chemists, but then modeling and structure is also good, but maybe it's not the leading thing. What are the new advances in modeling and structure and in thermodynamic stability of the interactions and different components? I think that is, from my perspective, part of the future.
And Kevin, maybe switching over to you, how would you optimize the interaction? This is something you deal with a lot, almost every day. What if you had a wish list, would you want to change or maybe do a little bit better as we're fashioning the Therapeutics Initiative?
Yeah, Mark, I can't help but take a step back and think about how extraordinary it is that we're actually asking the question in this way. And if you rewind 10 or 15 years, when I entered this area, when anyone spoke about biotech or academic startups, you cannot go 10 seconds without someone uttering the valley of death. The dreaded valley of death was this seemingly unbridgeable gap between where academic science ended and where investor interest or industry interest started. And when is the last time anyone on this call heard anyone complaining about the valley of death? So it's a really, really wonderful and very cool thing that we have all of these initiatives, and the reality that academia has built out of necessity, I think, a lot of the capabilities to move work farther along, and we as investors have gotten bolder and bolder to go earlier and earlier. So now the problem is, how do we optimize where those Venn diagrams overlap a little bit? So it's a cool problem to have, I would say. To me, I wouldn't change anything dramatically at this point. To me, it's a matter of figuring out how do we just increase engagement, because I think there's an incredibly beneficial two-way street that can occur. And it's a bit self-serving for me to say as an investor, but I think it's critical that engagement with academia, happen as early as possible. And from our perspective, we really welcome the opportunity to learn about cutting edge work, very early on, so that we can take the time to make sure that we truly understand it, the novelty, the potential impact. For some opportunities, this can be a matter of weeks, but for some, it can really be a process that unfolds over many months or even years as we track a certain area and educate ourselves on a new area of biology or technology. For the PI, we hope that there's a couple of benefits. The first is getting hopefully critical input on what are the killer set of experiments that would provide the data to position the work for further investment or licensing, if that's the route. There's often a broad choice of therapeutic applications, and we in our network of advisors can hopefully provide some guidance on what are the right animal models to set up, et cetera. Equally, and maybe more important though, what I would stress is the relationship aspect. So if you're an investigator thinking about potentially starting a new company, you're thinking about entrusting what is often a decade or more of work in your lab to a group of investors and executives that will be responsible for the monumental task of trying to translate that and get that technology into patients. So having many early actions where the discussion is more on a personal level, more about science, less about deal terms, financing, et cetera, I think is incredibly important if you're going to trust some strange person like me with your precious work. So and the last thing I'll say is, we've talked about, Mark, there needs to be a balance, because we all know that there's a special magic that happens in academia and happens nowhere else, and we want to be careful not to try to intervene too deeply. It's a bit quantum mechanical, and even that by observing it too closely, we risk sort of fundamentally altering the system. So I think our goal is to find these areas of complementarity, but avoid being meddlesome at all costs.
An excellent transition, talking about uncertainty principles. Yes. So I think in fact, it's something I'm always very, very passionate about, even going back to my industry days. It's how long do you let a really, really interesting and novel idea incubate before you try to make it practical? And I don't know that collectively as an industry or academia and industry, we've quite got it right. I think we dance around it, but it's definitely something that is always worth thinking about, and how we would make it better. Linda, maybe if you could give us a couple of comments from your experience, what would you want to add to the wish list of optimizing what we're doing?
Well, and this is something that has come up. I'm on the Stanford Board of Fellows for Stanford Med, and there are always so many ideas coming out and science projects that could turn into something interesting. But a lot of the folks who are developing these technologies or these, whatever, these chemical entities, they don't want to leave their day job. They want to stay in their role. They want to stay in research, but they also want to have at least some involvement with a company going forward. And so one idea that I proposed for Stanford, and they're doing a similar program where they're really trying to figure out how to get technologies out of the university, and would be to formalize a program between the investors, especially locally. Well, now, not so much now, COVID, it doesn't really matter where people are, but to really start the dialogue, and to Kevin's point about how we can really build up relationships between the investing side and the universities, so that there is this back and forth and people get to know each other, and they gain trust on people that they might be handing their baby over to, and keep them involved, and to try to, it's almost like a matchmaking opportunity. And I think that's something where I think, especially if the funds knew that there was this opportunity in this channel, that they probably would encourage more former entrepreneurs, or people who've exited or whatever, if they're looking for their next gig, to bring them on, and knowing that there is this opportunity to partner them with academics and really push forward some excellent science that's been done. So that's just one idea that we came up with at Stanford.
And I think, Linda, just to build on that point, and something that's been really a cornerstone in our thinking about the Therapeutics Initiative, is try to make this an extra opportunity for someone that's taken on an academic career. It shouldn't have to be a choice.
Right.
And can we create conduits by which you can do both, and do both successfully? All right, so we've got 2 minutes left. So Tim, you get the last word. You've done this several times. So bring us home in 2 minutes. What advice would you give from having done this successfully a bunch of times?
Well, first, what Linda said, I think she's talking about the SPARK program at Stanford.
Yep.
It's a very good program, and we should carefully look at that model. I think that we have to also somehow make or identify academic research more carefully, because when industry tries to reproduce it, they can only reproduce 70 or 90% of the publications that seem to have some pharmaceutical impact. And I think it comes, people ascribe it to lots of things, but I think what it comes down to is investigators need to be modest, and we need to be able to discard our hypotheses when the data shows that they don't fit. And I think that's the key thing. I don't know how we can do it, but academics needs to do a better job of controlling the quality of what is done. George, you figure it out. I can't. So there I go. So Mark, that's all I'd like to say.
Okay, I think that I'll maybe close the panel with one additional comment to something that Tim and I had brought up before. It's something I'm very passionate about, but if you took a step back and you looked at collectively the ability we have as the Harvard community, trying to find ways that we don't have to be expert in every single thing, even when we haven't yet partnered with an external entity, is something we have to pay some attention to. So I think that the ability to share resources, share expertise between our institutions, do it in a way where we can get that science hardened and take advantage of each other's expertise, I think is another way we can start moving towards what you were saying, Tim. So with that, first of all, thank you to the panelists for an excellent conversation and for all of the excellent advice. I'm sure I will be reaching out to lean on all of you to figure out what we're going to do as we go forward. And it now is my pleasure to introduce Mr. Len Blavatnik, who will be giving the closing remarks to close out the symposium for the launch of iHub and the Translator. Mr. Blavatnik.
Hello, everyone. Well, I guess they canceled my video, so you cannot see me from what I can tell. But hello from.
We have to try to fix that.
Now I can. Yeah, now I can. I see it. I think. Yeah. Okay. Hi, everyone. George, I see a nice bed behind you.
My wife gets the office. I get the spare bedroom. So.
Yeah. Greetings from London, where although there are a lot of new cases, the schools are open, the restaurants are open. My 16-year-old son is happy to be in school, so life is going on. But, it proves the point that we need better drugs sooner, faster. And, I was very impressed with this session and the panel. Thank you. Of course, thank you to George, Mark, and Tim. And it's really, from my standpoint, fantastic to see the team George put together in a relatively short period of time, and all the progress you're making. I think this type of effort, the Institute, and just a session like this, it shows, how much you kind of move forward in the whole concept of interdisciplinary collaboration in research and implementation, which is the key, obviously, to modern science, right? And I always thought that there's not enough of it at Harvard and in other universities. That people are in their silos, but I think you're really making great strides in breaking down the silos, and people are working together. And the last panel, in general, this session is fantastic. And I wish, and I congratulate all the recipients of the first award. A very impressive group, and of course the graduate students who are involved. And I couldn't be happier to see the results of my philanthropy. So thank you, everyone, and hopefully more to come soon.
Great.
Thank you.
Thank you so much, Len, and thank you so much for your support. And we're proud to show you what we've built so far, and we look forward to an even more successful future. And thanks to Mark Namchuk and the panelists and the graduate students for phenomenal presentation. Until next time, be safe.
Bye, everyone. Thank you.